Vanakkam. Welcome to this course on biomedical instrumentation. In the previous set of weeks, in the previous couple of weeks, we have been looking at ECG and EEG and EMG in very good detail. We went quite a bit deep, or we did a deep dive of these topics, as these are critical parts of any biomedical instrumentation course. If you are a biomedical engineer who has done a biomedical instrumentation course, you should know these topics in depth. That is an expectation. In this week, we will look at a couple of EXGs. EXGs means EXG where X is equal to R or O. What is ERG? That means ERG and EOG. What is ERG? ERG is electroretinogram and EOG is electrooculogram. So, with regard to that, we will build a foundation of the anatomy and physiology of vision, or the electrophysiology of the eye. So, in this video, we will look at the anatomy of vision, parameters of the visual apparatus in the eye, and the electrophysiology of the eye itself. So, the following are the light transmitting regions of the eye. First is the cornea, which is the outermost layer that is located here. It is marked here. That is the outermost layer, that is the cornea. And then there is the anterior chamber or the aqueous chamber. Between the cornea and the lens, there is the anterior chamber that is containing the aqueous humor. Right. This is a transparent fluid that is found in the anterior chamber, and then you have the lens, which is responsible for changing the refractive index. Lens as in like any other lens, the natural lens of the human eye. And then you have the vitreous chamber, okay, containing a different liquid, which is called as a vitreous humor. Okay. So, it turns out that each of these 4 light transmitting parts of the eye contribute differently to the refractive index. All of these contribute to the refractive index, except most of the contribution comes from where? Students would think that most of the contribution comes from the lens, but actually most of the contribution actually comes from the cornea itself. Okay. It's a common misconception that students think that, but all of them contribute to the refractive index, the change of the this. So, the anterior chamber contains an aqueous humor. It essentially provides a medium for transporting nutrients, also has some specific optical significance. The details of this is beyond the scope of this course, but I will touch just enough so that we can understand ERG and EOG. It turns out that it maintains a steady intraocular pressure of about 20-25 millimeter mercury. How is this maintained? Because the aqueous humor is getting recycled quite often. It's a really small chamber that is getting recycled once every 1.5 hours or so. So, the flow or the balance between the genesis, the creation of this fluid, and the draining of this fluid essentially creates this pressure difference, which is about 20-25 millimeter mercury. The vitreous humor is present in the vitreous chamber, which is posterior to the lens, and it helps to maintain the shape of the eye. Retina itself, this is the retina, that is the retina. It is not shown because of this color. This is the retina itself, and it contains various types of cells. Critically, it contains the photoreceptors. What are the photoreceptors? We know the photoreceptors, rods and cones, right? Those that are responsible for detection of light. And there are other cells, that these are essentially interneurons, horizontal cells, bipolar cells, amacrine cells and ganglion cells. So, these ganglion cell axons, they essentially form fibers that essentially sweep the entire inner retina and converge at the optic disc. So, that is the optic disc. Okay, again it is not showing, that is the optic disc. And it turns out that there is a convergence that happens between many photoreceptors to a fewer ganglion cells. Within the central nervous system, there is always this concept of convergence and divergence. When lot of information goes to a specific neuron, essentially that neuron acts as a synthesizing, processing neuron. There is what is happening is convergence. In many cases it need not be a single neuron. In many cases what would happen is that you would have 10,000 different receptors or neurons that converge onto say 100 neurons. So, in a 100:1 fashion. That means that there is convergence. That does not mean that every receiving neuron is receiving exactly 100. It means that there is a grand convergence of information that is happening. That is what it means. It is beyond the scope of this course for me to teach this, but I have discussed this in my other courses on classics in neuroscience and neuroscience of human movements. In fact, in neuroscience of human movements, I discuss convergence and divergence in the motor cortex and in the cortical region in great detail. Something those who are interested can check in YouTube. So, there is a convergence that happens. What that means is that these neurons, that is the ganglion cells, essentially act as synthesizers, those that provide or process information, receive information from multiple neurons and then pass it on to the next level after some processing is happening. And the visual signal itself goes through what are called as first step is photoreceptors, those that detect light itself. Those are the rods and cones. Then there are the bipolar cells, which are essentially interneurons, those that relay signals that come from photoreceptors to the next level. And then horizontal and amacrine cells are also interneurons, those that refine and integrate signals. And then ganglion cells essentially generate the action potentials that travel to the brain via the optic nerve. So, the processing of visual information happens not entirely in the eye, a little bit of processing happens in the eye, but most of the processing happens in the occipital lobe, in the visual cortex, in the brain. That is where it is happening. So, that means that this information will have to go to the brain. So, we have already seen this, let us move on. And then there are two types of resolution. One is spatial resolution, how much space or how much distances can you resolve, that is spatial resolution. The other is spectral resolution, whether you can distinguish colors, contrasts, that is a spectral resolution. Note that not everybody has the same amount of spatial and spectral resolution. In fact, there are several people who are color blind, lot of people who are color blind. Besides, also note that humans cannot detect all patterns and cannot detect all colors. Actually, this is a huge topic beyond the scope of this course, but those who are interested can check. Birds, for example, have unique patterns in their feathers and in their body that are detectable only by other birds of the same species, maybe of some other species, but that we cannot detect. What appears to be uniform to our eyes actually has intricate patterns that our eyes cannot detect. There are these kind of miracles in nature that our eye cannot actually detect. So, that means that something to keep in mind, that we do not see everything, but we believe everything we see. That means that there are things that happen truly that we do not see. That does not mean that it does not happen. So, something to keep in mind, but that is philosophy. But keep in mind that our eyesight, our vision has limitations in terms of spatial and spectral resolution. But not just that, there is more. in terms of adaptability, eyes can adapt to illumination levels from 1 to 10 to the power of 9 to giga levels. That is a huge range, a very broad range. Of course, this adaptation is not quick. This adaptation is relatively slow, especially in low light conditions, and these rapid changes cause what are called as dazzles. Usually, there is also a distribution of the photoreceptors found within the eye. Cones, those that are specialized for photopic or color vision, are found highest in fovea, and their density, their distribution decreases as you move peripherally. As you move away from the fovea, the distribution of cones reduces, whereas rods, those that are responsible for night vision or scotopic vision, have their max density in the periphery and less in fovea. So, there is a, it seems like there is a clear work distinguishing work, it seems like there is a clear work distribution rather. So, note how much sensitivity you have. A dark adapted eye can detect a change in power of up to 1 picowatt per centimeter square, which is huge, and a single rod may be quite often excited by just 1 photon, which is amazing. A single photon may be detected by a single rod. So much is the sensitivity and the visual acuity of the visual system. But note that although individual rods respond to single photons, to be perceived, because there is I already mentioned, there is neural convergence. There must be information coming in from several photoreceptors for the ganglion cell to activate the optic disc. That means activate at the optic disc and further processing. That means there must be several rods that must be activated for you to be able to perceive. I did not say that 1 photon may be perceived. I said 1 photon may be detected. 1 photon is detected by a rod, but several rods must be simultaneously active for you to be able to perceive. There is a difference between the two. Please note that. So, what is the activation energy in 1 photon? About 3.9 into 10 power minus 19 joules. That can be detected by the rod, which is phenomenal ability, phenomenal acuity. Something to keep in mind. About 5 to 10 rods must be activated for you to be able to perceive. Something to keep in mind. Spatial resolution is affected by cone distribution, or how much cones or cone spacing is what determines the. It's about 2.5. It's about 2.5 microns in the fovea, whereas spectral resolution depends on the trichromatic vision. There are 3 specific wavelengths to which you will have peak sensitivities. Now, what are these wavelengths? Find out. I will leave this as an exercise for you. What are the wavelengths at which you will have this color vision or the spectral resolution? What are those wavelengths that affect the spectral resolution the most? Note that modern digital cameras, modern high-end cameras essentially struggle to meet the high dynamic range of the eye. So, in HDR mode, in high dynamic range mode, what modern cameras do is they take multiple exposures to come up with a single picture to have the dynamic range. But this is taking multiple pictures, and in various times, multiple exposures. But the eye has a different mechanism. What the eye does is it has cells that are specialized for different purposes. So, it has a single exposure, but all of this is detected in a single exposure through specialized cells. So, there is a difference in the mechanism, between the high-end cameras and the eye, but even with the highest or the best cameras, they still do not, are almost struggling to meet the dynamic range of the eye. Anyhow, let us move on to the electrophysiology. We are interested in measuring the electrical signals from the retinal cells, which are photoreceptors and ganglion cells, to essentially assess the health of the visual pathway. We are interested in finding out what is the health. So, by the way, the retina has a multi-layered signal processing pipeline, an excellent, extraordinary signal processing pipeline. Note that the retina and the eye are part of the, they are not just the visual system, but they are essentially an excellent signal processing or the part of the brain processing system. So, it turns out that in darkness, photoreceptors are electrically active on at a constant dark current. Light typically causes hyperpolarization, reducing the dark current in proportion to the light's intensity. This is quite unusual in neural processing. Usually what happens is that whenever there is a signal that is detected, it causes depolarization, changing the frequencies, but in this case light actually causes hyperpolarization, reducing the dark current in proportion to the intensity of the light, and this essentially generates an action potential that is then sent to the brain via the ganglion cells. So, and it turns out the frequency of this action potential, which is essentially a form of analog to digital conversion or rate coding or frequency encoding of the intensity happens. So, not the amplitude, but rather the frequency of firing of the action potentials of the ganglion cells essentially encode the light's intensity. So, a form of analog to digital conversion. But why do you need this? Why do you need to perform this analog to digital conversion? Because this visual information will have to travel a long distance. How much distance? Few centimeters, perhaps more than 10 centimeters. No? The distance between my eye and my occipital lobe, which is here, which is in the posterior part of the brain. That distance is perhaps more than 10 centimeters depending on the head size. That is a long distance in neuronal terms. 1 centimeter is a very long distance. 1 centimeter is 1000 kilometers in neuronal terms. So, it has to travel a long distance to ensure that the signal to noise ratio is minimal and the transmission is faithful. You perform this analog to digital conversion, essentially performing this frequency encoding or rate coding of, it is a deep concept of neuroscience, not having the time to discuss this, but do check my other courses in which I discuss what is rate coding. Okay. Rate coding means essentially frequency encoding of a particular intensity. This applies to various, this is a very common phenomenon in the neural transmission system. This happens in many other systems, but critically the difference is that light essentially changes hyperpolarization, which then gets converted into the frequency encoding in the ganglion cells, is something to keep in mind. So, common clinical measurements of the eye or the visual system essentially involve electrooculogram, electroretinogram, multifocal electroretinogram, pattern electroretinogram and visually evoked potential. In this, we will restrict our attention to ERG and EOG, and we will not be discussing the others, multifocal ERG, pattern ERG and visually evoked potential, which we will leave as an exercise for the students to explore by themselves. So, with this we come to the end of this video. In this video, we looked at the anatomy of vision and the parameters of human visual apparatus and the electrophysiology of the eye, and how the eye essentially generates these potentials, and how what are the various types of cells that are present in the eye. This is just a very brief introduction without going into the details, because our interest is not the physiology of the eye, but rather our interest is the diagnostic method, which is electroretinogram and electrooculogram. With this, we will end this class. Thank you very much for your attention.